Great. All right. Well, thanks everybody for showing up here virtually and inviting me to talk to you. It's a nice change of pace for me to be able to sort of speak to a wider audience about some of the stuff that I do. So I'm calling this here a tour of an ocean research facility. I'm going to kind of walk you through my labs and talk to you a little bit about the management of the labs and the technology and the science that we do there. So you got a bit of an introduction about me already. When I'm speaking to younger people, students, early career people, I think it's nice to kind of weave in a little bit of story of just my career and talk to you guys about career options in the field. So I started out at the University of Toronto and University of Guelph for my bachelor's and my master's. So I'm an Ontario guy. As Taylor mentioned, I worked at Environment Canada for about 10 years, again, in a technical role. And there I was doing microbiology, chiefly environmental microbiology, looking at waterborne pathogens in lakes and rivers there, sort of beach closure issues and things like that. It's a good job, but it's sort of a job that stayed the same for a long time and kind of getting tired of suburban living and wanted to change. So an offer came up at Memorial University in Newfoundland, which sounded, exciting change. And it wasn't really, I'll say, directly in my field. I'm not a fish guy, ocean guy by sort of training. But I had a lot of sort of technical skills that were going to apply to this new facility that they were building there. So I applied, and I, to my own surprise, actually got it. So this is where it is. This is the Ocean Science Center. It's just above St. John's in Newfoundland. It is primarily a research facility, and the teaching is done on campus down the road. And as you can see, it's situated on the ocean, which is very handy for keeping sea life alive in tanks inside. We can pipe that water in. Actually, I don't know if I move my mouse around, if you guys will see that or not, but I won't worry about it too much. So we were able to pump the water into our fish tanks at the different buildings. That's a lot easier than having to create seawater by adding salts for inland facilities and using that water, like you might for a home aquarium or something, or you have to replace the water, and you have to be really particular about water quality. Here, the water comes in, goes through a tank, goes out, and we don't have to worry about chilling it because the ocean here is quite cold. Just a little bit of background about the university. I'm going to be calling it MUN. That's just our sort of short version for Memorial University of Newfoundland. We've got, I'm not sure, we got like 16 or more faculty at the Department of Ocean Sciences. They do everything from fish physiology and oceanography, and there's a big group that is studying aquaculture, which I'll talk some more about later. And we're a pretty special place just because of our facilities. So to support the research, we have a lot of these aquatic facilities, the tanks that I had mentioned, that can be heated or chilled or freshwater or flow through seawater. We have our own dive team, divers that can go out and collect specimens for research or teaching, as well as do scientific diving for, transects and things like this. Most of the faculty will have their own specialized labs, and they'll also have common use labs, where typically the more expensive equipment is kept, or the more specialized equipment is kept, and that's kind of where I fall into, is in managing some common use places that include some of the analytical instrumentation. So that kind of brings it to me again, and what I do as a lab manager, and what does a lab manager do. Most universities, research facilities, and I suppose industry too, have these centralized research centers, like called a core facility or scientific platform, where they'll have hired technicians and staff that are especially skilled to run this equipment. And as a manager, I coordinate all that, and I help the scientists with their experimental designs, and making sure all the equipment is in good working order. We will train students and staff so they can also learn how to use the facilities. And you may find some managers who are very specialized, particularly like in medical centers or bigger cities where they're basically just having maybe a facility full of microscopes, and that's what they do, and they do it really well. Or there may be more people like me who are in smaller facilities that just have kind of like one of everything. And you have to be a bit of a generalist. You have to kind of dip your feet into medicine and ocean science and engineering and all sorts of different techniques. Of course, we have a lot of paperwork as well, just the various aspects of maintaining lab safety and certifications, finances, and all that, as well as there's a bit of a role as a business person, because like most places, you're always looking to how to fund all of this, and it's a very expensive sort of operation to keep going. So what I've talked about is kind of the framework of my presentation today. I'm going to use a discussion of our technology, which is really what I, in a way, am passionate about. I really just kind of like the gear, the equipment of science. So I'm going to talk about a bit of microscopes or scanning electron microscopes, some of our deep sea equipment, and containment zone. So some windows around here so you can see my screen here. And then I'll also give from each of these an example of what is the actual science that we're doing with this. So I'll talk a bit about microplastics, zoology, oil spill research, and aquaculture stuff. And I will not talk to you about administration, because that is the least fun part of the job. All right, so this is an overview of the Cold Ocean Deep Sea Research Facility at CDRF. So the picture up in the right there, that says the building on the right, and it has what I think of as 4 main kind of operational units within it. So the containment zone, I'm going to talk about most of these in detail later on. The containment zone is where we can study fish diseases. It's specially designed basically like a biohazard facility so that we're able to study living pathogens without risking releasing it to the environment. The deep sea labs, maybe not what you imagine, what they are basically pressure vessels that we can use to recreate the conditions of the deep sea, chiefly the high water pressure that you would find there. Histology and bioassays. Histology is essentially the art and science of preparing tissues for microscopy and pathology, creating slides, and we do other sort of assays at the bench, a lot of it for like environmental monitoring at the offshore oil and gas. I'm not going to delve into that too much today. And then lastly, we have microscopy and cell biology. So pretty much like it sounds, microscopes. Seems to change very. Okay, so let's start off with the deep sea. And I don't really have a lot of feedback here in terms of seeing anybody or anything. So if there's any questions or issues, maybe Taylor, you can. Oh, so the deep sea. So in the deep sea labs, as I mentioned, we have these chambers. We have 2 of them. They'll hold 19 liters of water, and there's a pump that will pressurize that. So using that pump, we can create the pressure that we find in water that is 3 kilometers deep, so fairly deep. And what is special about these is that we can actually continuously flow the water through those chambers while we're maintaining that pressure. So it's not just a bottle that you close and pressurize. We can actually keep things alive in there. We can replace the water and the gases. You can heat and chill it. What's sticking out of the top here is a camera. Observations as well. This is just the view from the top of the empty chamber. So we can alter those water conditions to, for instance, simulate ocean acidification by changing the pH. We can do tests for toxicity. So that brings me to my first example of some of the science we use this equipment for. I think everyone is familiar with the Deepwater Horizon spill of 2010. That was a rupture that happened, I think it was 1,600 meters below sea level. Generated, for better or worse, it generated quite a lot of science about spills and oil contamination. And one of the kind of notable things about this was the observation of oil settling out in the bottom of the ocean. You kind of think of oil as something that floats, and most of our responses to oil spills are at the surface. But there are different processes that can allow that oil to sink. When it's been out in the sun on the surface, the light will evaporate. Some will evaporate off the lighter components, and some of the heavier tarry components can sink them. And some of that can also bind up into materials that will sink. So we have a professor, Dr. Ida Passow, is known for studying heat snow. So this picture on the lower left is these knotty things that are kind of agglomerations of detritus and organic matter, and they can bind up the oil as well. It's called snow because it slowly sinks down to the bottom of the ocean. So, the deep sea is an interesting topic of research. It's most of the ocean, when you think about it. And it used to be thought of as a really kind of a barren, desolate place, but we're really discovering there's quite more diversity of life there than we've originally thought, and it's a very unique life. It is difficult to study, as well. We don't know a whole lot about it, mostly because of this high water pressure that will crush us if we try to dive too deep, and we have to have special diving gear. And even in the lab, it's very hard to recreate those high pressures, and that's what our equipment is for. Consequently, when it comes to like oil spills, most of what we know about the toxicology of that and how it affects life is based on research that's done on atmospheric conditions in the shallows of the Gulf Coast and such. And that may or may not really apply to what happens at depth. So here is one of the pilot projects that we had done in the deep sea labs, which was really starting at the beginning and asking some basic questions of how does oil degrade in the deep sea. There are bacteria that will naturally degrade oils, different kinds of oils in the water. That is, one of the responses to an oil spill is to just let it get eaten and degrade. As well as doing things like adding surfactants that will break up the oil. So it's more bioavailable. It's easier for bacteria to degrade it. So for our experiment, we took some crude oil and some of the natural seawater here with its bacteria in it, and we put it in our chambers, and we pressurized those chambers to be 1,500 meters worth of depth. And we took another bottle and we just left it at atmospheric temperatures, and we both, I think, put them to around 4 degrees Celsius, which, at least in our waters is, the temperature is as close as we could get to the temperatures of the water at that depth. So the good news was that the oil did degrade. This is these charts on the right. This is kind of a fingerprint of the different chemicals in that oil. And you can see after 7 days, we've lost 49% of the atmospheric pressure, 0.1, and 40% of the water at depth. When you get down to 35 days, 81%, most of it was gone at atmospheric and less was gone at depth. So it may be slower degradation at depth, but it does seem to be going towards completion. We've used this equipment for other similar sort of studies with oils, looking at the interactions of these surfactants I mentioned with the oil, if there's any changes in how those droplets are formed under deep sea conditions. And what I'm keen to move into more now is really deep sea toxicology, actually taking some animals, zooplankton, and that sort of thing, and seeing how they respond to toxins in a deep sea condition. All right, moving on. So we'll be moving upstairs here our microscopy and cell biology lab. So we have a lot of nice equipment in there, including just sort of the workhorse of many biology labs, the compound microscope or light microscope. So shows up here, but so I'm sure most of you guys are familiar with this. You have a light shining through a condenser, which focuses the light onto your sample, like on the slide, and then the objective reflected by various means into your eyeball or camera. The objectives are sort of most important and expensive part of these microscopes. And for anyone who's into photography, might transition quite well into microscopy because many of the principles are the same. Understanding what aperture does to your image and different coatings and things like this that you have on lenses. That's why these lenses can be, in your educational or in your classrooms, maybe they're a few $100s, but in research labs, they're often thousands or even tens of thousands of dollars for high-grade optics for microscope. So like I said, we would use these on slides that we prepared and stained so they can be seen in visible light. This is, on the bottom right is a picture of gills. So in order to look at tissues, you have to section them very thinly and then add the color back with different stains. So this is a very thin section through the gill of an American place that we would look at for any abnormalities associated with exposure to toxins, for instance, at offshore oil and gas platforms. Trouble changing my slides here. Okay. Now the another type of microscope or another type of light that we would use is fluorescence. And in this case, it's different than light microscopy. You're actually making your sample glow, and in a very specific wavelength. So you could be adding different kinds of stains that bind to specific types of tissues or specific types of proteins or other kinds of probes that will do this. And you're using a laser light or a filter to excite the fluorophores in a very specific wavelength that then glows back and is filtered until it comes back to your eye or camera. So in these pictures, they both have been stained with dyes that will fluoresce, that will make the DNA fluoresce. So in this larval cod, basically the whole thing is glowing. There's DNA through it. And then these cells, these blue dots, are the nucleus of DNA. So as I was saying, that's really useful because it allows you to distinguish different components of the animal or the cell. You can use it to target very specific things like specific bacterial pathogens. I mean, you probably be seeing a lot of scientists developing probes for COVID-19 virus right now. And it allows for higher resolution methods in microscopy. This picture is an example, which I'm going to kind of lead into, of a fish that has ingested plastic. I've purposely fed this zebrafish plastic that fluoresces when I excite it with the right wavelength of light. So I can distinguish the plastic in the gut of this fish from something else that it might have eaten. So microplastics as an example of microscopy in their labs. I would assume that everyone's pretty familiar now with the issue of plastics in the environment. I think a lot of people think of this bird on the bottom left as the problem of birds and fish and marine mammals eating chunks of plastic, filling up their gut certainly is a problem. But it's really a problem throughout the scale of life. Some plastics are small. We typically would call microplastic something less than 5 millimeter in size. So you have fibers that come off of your clothes or microbeads that were used in soaps and things. But also all of this larger plastic will break down into smaller plastics, such as up in the picture in the left. So what I'm interested in is how these microplastics impact the sort of smallest animals in the ocean, which are the zooplankton. These are really key to the web of life. Most fishes are dependent on eating zooplankton or going up through the food web. These are little crustaceans and copepods, things, shrimps that is what's filling the ocean, playing that important role. And they too can ingest plastic. So this isn't my work. This is some work from a few years ago where they demonstrated the capacity for zooplankton to ingest plastic in the lab with these fluorescent plastics. Zooplankton are generally fairly indiscriminate feeders. They will generate currents of water to bring particles into their mouth or filter feed with membranes and different methods. And I don't really believe that they can distinguish plastic from food. And these are really small plastics, 30 microns, or less will get ingested by them. And now it's kind of a question of what's the impact of that. Don't think the impact is truly understood. We know that it's going to have an impact on the feeding if they're eating plastic instead of food. That plastic is nearly indestructible, so it's just accumulating and being excreted and being ingested. And if it ever gets to the point where the amount of plastic in the ocean is, competitive or in numbers to food, then that's a wasted effort. As well as some plastics can absorb toxins and they have some toxic effect themselves. That's something that's kind of actively being investigated. It's really hard to detect plastics of this size in the field, although there's been some progress on that. When you're looking at that under a microscope, determining plastic from a bit of a shell or something like that, this is not easy to do. So that's why these laboratory studies are using these kind of artificial fluorescent plastics. So I've done some work with rotifer, which is a zooplankton that we actually use to feed larval fish in an aquaculture setting. I'm going to guess this may not show up very well. I'll have a little video, give it a shot. So corona, there's these hairs at the mouth that continually beat and create this vortex, this current that brings in the particles as they swim through the stomach. So they run around, swim around, and pick up everything. And in the experiments I do with these, I would be adding plastic at like a microgram per milliliter. And that's kind of roughly equivalent of if you took this bottle cap, and you ground it up like finer than sand into just the finest dust, and put it into a 5,000 liter tank. So it's not a lot of plastic, but when it's broken up so much, it's kind of everywhere. So a smaller animal, like the smaller animals like zooplankton, are just bound to encounter it. And again, throw this video up here. I'm not sure if it's going to come through or not, but the picture on the right, you can see here are just some of my own pictures of this plastic, this fluorescent plastic that I've added to the water in experiments, glowing within the basically transparent rotifer. And on the left, there's a video of them swimming around, and you might be able to see the little dots from the water. Okay. So in the same microscopy labs, we also have a scanning electron microscope. So when you want to get to much greater resolutions, there's actually just a physical limit to what light can resolve. And that's what's being shown just in these red pictures here, where on the right, even though there's two individual peaks, just the wavelengths of light will cause them to merge together. That's resolution. So one way to get further into that is to use an electron beam, which has a much greater resolution. As a wave, it has a much smaller wavelength. And instead of using glass, you're using magnets to direct the focus of the beam onto your sample, and then it emits the electrons back into your detector. So greatly improved resolution. You're no longer using color, and it also requires some fairly specialized sample preparation. Basically, the sample has to be very dry, or else it'll kind of vaporize in this electron beam. This is our SEM. If you guys see scanning electron microscopes in other labs, they're probably going to be much larger collections of tubes and apparatuses and stuff. They started to miniaturize these like other things. So this will give us like 100,000 times magnification. And we get into the realm there where you can not only see bacteria, but you can see like the surface features of bacteria. Although in our use, more typically, we are looking just for better pictures of like marine invertebrates and parasites and some of the early life stages of So I'm just going to play a little game here instead of going too much into the research of what we've done. I'm going to throw up a few pictures, and I do not see if I have access to the chat window while I'm in presentation mode here. I don't think so.
Stephen, I can always kind of let you know what they're saying or what they're guessing if you want.
Yeah, sure. So if you guys wanted to throw in the chat what you might guess that is, and Taylor can recite some back, and won't spend too much time on it, but let me know. Any guesses?
Not yet, so.
No. Okay. So the size of this is like 400 times magnification. You would be able to see this under a light microscope, but you wouldn't get the details of like these little spikes, for instance. This is a woodlouse foot. So the foot of these insects, different names for them. That's what that would look like under a scanning electron microscope. So, kind of cool. Here's another slide. It's pretty hard to guess, I guess, if you haven't really looked at these kind of images before.
Oh, we got one guess from Caitlin.
They do look very feather-like, but it's not a bird. Something very small. Butterfly scales. Yeah, that's very close. This is scales of an insect, a mosquito. So on the back of the mosquito, when you get it under the SEM, you'll see these scales. I didn't even know that. I was just sort of trying different things out when we first got it, and I was really quite to see what these scales look like. All right. Number 3. This is actually something I did show you a different picture of before. It's not very high magnification. From a fish. Gills, yes. So the gills of a fish are really evolved to maximize their contact with the water to take the oxygen in. So we have the filaments that are projecting out, and on the filaments are lamellae, secondary filaments. That's what these lobes are that are sticking out. So I showed you earlier like a cross section on the slide of branching features, and that's what it looks like in a kind of more 3D view. Okay, last one. This was also one that was pretty surprising to me when I first looked at it. The specimen is kind of dried out. These grasping claws. I guess I wouldn't think you would guess this unless you had an idea of what I'm talking about later. So what this is sea lice. So this is going to segue into later talk, part of the talk. This is a parasite of salmon, salmonids, and it attaches onto the skin with these hooks, and then at its mouth feeds. So you're going to the underside, and it has this cap on top. Sucks the blood. So you get these kind of cool details that you wouldn't be able to see on the light microscope using scanning electron microscope. All right, so I'm talking about parasites, about the containment zone of the CDRF. So this is where we do disease challenges. It is a part of the facility that's specially designed and certified for us to work with live pathogens. So this is an aquatic containment lab. There's 3 levels of aquatic containment labs, and we have the highest level, certification 3. So we can basically work with any aquatic pathogen in the world. So there's going to be ones that are typically relevant to this part of the planet, and bacterial, viral, and parasitic pathogens, particularly those that affect the aquaculture industry. But we could also look at emerging pathogens if there was, something that we saw that new in the area, or might be coming from Europe or somewhere else. We'd have the capacity to do that. The facility is designed to treat the water. We have a wastewater treatment facility here, and everything is kind of monitored very carefully so that we don't accidentally release these pathogens back into the natural environment. So that allows us to do things like tests and develop vaccines, different types of feed that would be used in aquaculture that might help resist disease, other kinds of drugs as well. There's a collection of wet labs in here that we can work. A lot of the operation of this is training. It's just sort of personal hygiene, which we're all very familiar with now with the pandemic in terms of PPE and not doing things that carry out the diseases or into an experiment. So I'm going to talk a little bit about aquaculture. And just as context, it's kind of an important topic for Newfoundland and Labrador. It is growing industry here that's focused on salmon, Atlantic salmon, particularly on the south shore of the island, as well as blue mussels. There's quite a big push from the government to grow this industry. These numbers are now, but from 2014 to 2015, 61 million. Some ups and downs since then. So Atlantic salmon farming goes through processes of hatching the eggs through Galvin and fry to smolts, which are then transported to sea pens. So these are large nets, basically, that are in the open ocean, or the coastal ocean, where the salmon can be fed and grown out to the right size for market. There really isn't any use of land-based recirculation systems, which is the other main method of aquaculture in Newfoundland outside of research labs and maybe smaller scale kind of specialized operations that are trying to grow, like a tropical fish or something like that. So maybe just as a sort of topic for conversation, we can look at some of the pros and cons. And it's sort of a very interesting viewpoints on aquaculture across the country. People in BC may have very different views than people in Newfoundland, for instance, just generally speaking. And as a kind of an emerging industry here, it's an opportunity to do it right. So some of the pros, as I mentioned, as I'm sure you guys are familiar, the fishery had collapsed in Newfoundland and has really gutted our rural areas. So people see this as an important opportunity for those rural areas to regain work, since these farms are going to be in the rural coastal areas of the province. And by using the farms, we are replacing some of this lost product, this lost protein that we would have gotten from wild stock. They're unlikely to replace. And maybe in an optimistic viewpoint, you might reduce some of the pressure on the wild stocks. These charts are just showing some comparisons of fish to other meats. So one of the good things that's often touted about salmon is the amount of food you put in to grow a kilogram of salmon is less than pretty much any other meat. So you need, 1.2 to 1.5 times as much food as weight of the fish that you get versus cattle. You need to put in like 6 to 10 times as much feed. It's the feed conversion ratio to get a kilogram of cattle. And likewise for CO2 emissions, the belief is that it is a less intensive industry for CO2 emissions. And at the end of the day, it produces a very desirable product, salmon, which is a healthy meat and high value. So on the other side of things, there are certainly complaints or concerns about the environmental damage from these farms. We're using the ecosystem, as a service to the industry here, and instead of having to treat wastes from the fish, we're using the ocean for that. As well as if there are any drugs that are being used, there may be some impacts on the local ecology from the use of those drugs. Escapes are a concern. These fish are, bred to be fast growing and resistant to disease, for traits that are appropriate for aquaculture. So if they escape, they are going to interbreed with the wild population, and there may be, concern of diluting the natural diversity of the wild fish if there are escapes from storm damage or something to the nets. Right now, you still need to feed these fish, and you need to feed them other fish. So it's not as though you aren't lessening the impact, or you aren't completely eliminating the impact of a fishery. You're getting smaller forage fish to use as fish meal as a component of the feed. These farms are, the cities of fish. They are places where diseases can occur with greater frequency, and these are going to be the same diseases that would impact wild populations. And we've had, a few events here, really in recent news, where just kind of bad animal welfare or bad events where weather or ocean warming has resulted in large die-offs of salmon or other fish that are used at the farms, as well as some controversy about the industries trying to kind of circumvent with a government that's eager to grow this business, trying to circumvent some requirements. So I guess I'm more on the side of I like the idea of aquaculture, and I like the idea that it is amenable to improvement. So there is fairly active research at the Ocean Science Center into making aquaculture a very sustainable farming practice. That includes things like replacing the fish meal or at least the fish oil component of feed with vegetable oils, like camelina oil, um, as well as looking at alternatives to drugs that are being used on the fish. And there can be different components of the feed, cleaner fish, and vaccines, which I'll talk a little bit about. So it's back to sea lice here. Um, I think I kind of described this already. This is this parasite that attaches to the salmon. It is like naturally occurring in the wild. Wild salmon will get it as well. It can damage the skin, and in the worst case, it can alter the tissues so much that the osmotic balance is thrown out and the fish can die. We've used drugs in the past that have been added to the feed, but the sea lice are now becoming resistant to these drugs, and that's a common problem with drugs that are being used for pathogens, parasites, and such, is that they will in time are inevitably going to develop resistance. So the different kinds of things that are being proposed or studied or actively used to address sea lice are, well, you can just try to breed salmon that attract the lice or otherwise resistant to the lice. It's kind of a long-term project, and for that the lice may. There's been a lot of effort on altering the design of the sea cages themselves. The sea lice are typically on the surface of the water, so if you can shield the edge of the cage or sort of submerge the cage with just the tube coming up, as much sure you can create conditions where the sea lice won't be able to find and attach to the salmon. You can do bath treatments. So you can take the fish out of the water and put them into a wellboat or other cages that have fresh water or hydrogen peroxide, and that will cause the lice to fall off. That's stressful for the fish, but it's something that many farms might do a number of times in the year. There are even gadgets that will use cameras to see the lice on the fish and zap them with a laser. It sounds kind of incredible, but seems to work to some extent. And then there's kind of the two things that we've done the most of. One is to develop feeds that have different ingredients, different kinds of proteins that will stimulate resistant mechanisms, immune mechanisms in the fish. That'll give a kind of a percentage improvement to the fish's resistance. And the other one is cleaner fish that will actually are fish that naturally eat the lice off of salmon. So I'm getting to the end here. I'll just talk a little bit about these cleaner fish. So this guy is a lump fish. It's attached to my oxygen probe. They have these modified fins that kind of act as suction cups, and they're really slow moving fish. You could reach into the tank and grab one, but they can attach to rocks and kelp and things and ride out pretty much any storm that way. So they are grown at the OSC and are then added to the sea pens once they hit maturity. In the sea pens, they'll have set up kind of artificial kelp forests that would be a cleaner station where the salmon will come in and the lumpfish will come off and pick off the sea lice and eat them. So it's been quite successful. It's sort of still an active area of research and improvement. It can replace a lot of the drug treatments and reduce the needs for these baths. But because it's kind of a new species for aquaculture, a lot of the vaccines that all salmon would receive aren't going to work for lumpfish, and they can pick up other bacterial diseases just like any fish. So we can use our disease challenge facility to develop those vaccines. We can take it to the sort of final step of actually injecting the fish with a vaccine and exposing it to a bacterial pathogen to see if it works, which is what you really need to do before use it in the wild. So I think I've talked a good deal. Yeah, that's where it is. I think I hope that was an interesting tour through of the lab and some of the things we do. I hope you maybe got some perspective on how there are kind of different career options that engage in ocean science, and also I would say it's really worthwhile to think that your skills that may you have gotten from some other field could definitely apply to ocean sciences. It's not what I went to school for, but I've ended up there, and vice versa. So I will